for us to review the basic anatomy of the thyroid gland. So we know that the thyroid gland is butterfly shaped, and it has two lobes which are connected by the isthmus. Here we have the larynx, and here we have the trachea. So when you take an histological slide or tissue of the thyroid gland, you're going to see the following. So you're going to have a follicle composed of the cuboidal follicular epithelium and the colloid, which will understand their function in a few. Then, of course, you'll have a capillary to perfuse the thyroid gland. And you'll have the C cells, which are going to understand the functions later on. Now, for the functions, it's very important to understand that the thyroid gland is very important for formation of T3 and T4, which are thyroid hormones. So this is triiodothyronine, and this is tetraiodothyronine, also called thyroxine hormone. So the most abundant hormone to be produced is usually T4, approximately 90%, while T3 is usually only approximately 10%. Another important thing to really remember is that even though T4 is the most abundant, T3 is the most potent. So usually in our tissues, before thyroid hormones can function, T3 is usually converted to, no, T4 is usually converted to T3, sorry. Now, another function of the thyroid gland is for production of calcitonin, and this one is usually produced from the parafollicular cells of the thyroid gland. So we're going to understand the functions of calcitonin in our other videos or our other lectures. So iodine is very important for the formation of T3 and T4, and there it's very important that you understand the daily requirements of iodine, the weekly requirement, and the yearly requirement. So the weekly requirement, the weekly requirement is 1 milligrams per week, but yearly it's approximately 50 milligrams per week. So that's the amount of iodine required by the body for efficient production of thyroid hormones. Now, before I come there, we have the different steps of thyroid hormone synthesis. One, we have iodide trapping. And for iodide trapping, we always have something called a sodium iodide importer. So basically, this iodide pump is important to take in iodide ions into the follicular epithelium. So this process is usually affected by TSH, in that the moment your body produces a lot of TSH, then the rate of iodide pump is going to increase so that we can produce more thyroid hormones, of course, by taking in more iodide ions. Now, the second step will be oxidation of iodide ions. Now, after they have been taken into the follicular epithelium, we have an enzyme called thyroid peroxidase. Now, this enzyme is very important to convert iodide ions to iodine. And we'll see that hydrogen peroxide is usually very important for this particular step of thyroid hormone synthesis. Then the third one will have iodination of tyrosine and thyroid hormones formation, what we call organification. So after we had formed our iodine, it will be combining with thyroglobulin in different proportions. So when it does so, it is going to form DIT and MIT. So DIT is just diiodothyronine and MIT is monoiodothyronine. So in short, when I take two iodine molecules together with thyroglobulin, I'm going to form diiodothyronine. But when I take only one iodine molecule, I'm going to form monoiodothyronine. Now, after forming DIT and MIT, the next step should be coupling reaction. So in coupling reaction, you're basically taking different combinations of MIT and DIT to form T3 and T4. So for us to form T3, we need to have 3 iodine molecules, and that's why you combine one MIT and one DIT to give us T3. But for DIT and DIT, we are going to get T4. Now, don't be deceived because you have another hormone called reverse T3. Again, this is usually formed from the coupling of diiodotyrosine and monoiodotyrosine. Yeah, sorry, this was diiodotyrosine, not tyrosine, tyrosine, sorry. It's tyrosine because tyrosine is the major component of thyroglobulin. Now, reverse T3 can also be formed from MIT and DIT, but it appears that this reverse T3 doesn't have a function, or it appears to be non-functional in human beings. Yeah. Now, when you go back to this diagram here that I was skipping intentionally, we can see that first of all, we need to have the sodium-potassium ATPase, which is going to pump out sodium ions and take in potassium ions. So this is basically meant to lower the concentration of sodium ions inside this epithelium, while increasing the concentration out of the epithelium, so that sodium ions can move down their concentration gradient back into the follicular epithelium. So when sodium ions are moving into the follicular epithelium, they usually carry iodine molecules. So this is what we call the iodide trap of the sodium iodide importer. Usually it's a active transport which requires energy. Now, after iodine has been taken into the follicular epithelium, we can see that in this process here, we have this pendrin molecule. This pendrin molecule is basically a co-transporter where chloride ion is taken into the follicular epithelium while iodide ions are being extruded out. Now, importantly, these iodide ions are being converted into iodine. And the enzyme is thyroid peroxidase, which requires iodine peroxide. Now, after forming iodine, it usually combines with thyroglobulin in different proportions. I think I talked about it. But again, remember this thyroglobulin that we have is a protein because it's made up of amino acids. And that's why here we can see that we have tyrosine molecules. They are going to the endoplasmic reticulum, where we usually produce proteins. Then after that, they're being taken to the Golgi apparatus, where we have post-translational modification. Before now, they can be packaged to be used in the colloid. Now, after they have reached the colloid, the thyroglobulin will combine with iodine in different proportions. And this is what we call coupling reaction. So in that particular scenario, you're going to form MIT, monoiodotyrosine, and diiodotyrosine. Now, after forming MIT and DIT, the two of them can combine in different forms or in different combinations to form T3, reverse T3, and T4, as I've just talked about in a few. Now, all these MIT, DIT, T3, RT3, T4 usually attached to the thyroglobulin. But now, remember, after forming all this stuff, we need to take them back to the bloodstream because they're supposed to be used inside the body. So what happens, number one, we always have pinocytosis. Now, in pinocytosis, you're basically going to take this thyroglobulin together, the components back into the follicular epithelium. Then later on, you're going to see how lysosomes can be very important to come and help in lysis of this complex to produce T3, T4, then you release it. Let us go to the other videos. But before we go to what I wanted to explain, we have this organification now, just basically a review. You can see that we have mono plus mono forming tri, but again, mono plus di can form reverse, but di plus di can form thyroxine, which is quite important. Now, this is what I wanted to talk about, the release of T3. Now, we see that here we have our amino acids, tyrosine. They are very important for thyroglobulin synthesis. I think I've talked about it. Now, thyroglobulin will be taken into the colloid. And here we have iodine. Here we have the iodide tram, where we have the sodium iodide importer. Then you take in iodide ions. Then after that, they oxidize and they're taken into the colloid. So after being taken into the colloid, we can see that they combine with thyroglobulin in different proportions to form MIT and DIT, which now can be coupled to form T3 and T4. Now, after forming T3 and T4, they're going to be taken back into the epithelium via the pinocytic process. Now, in the epithelium, they'll combine, or they'll rather fuse with a lysosome to form a phagolysosome. Now, this phagolysosome contains some hydrolytic enzymes which are able to break down thyroglobulin, separating MIT, DIT, T3, reverse T3, and T4 from it. Now, T3, T4, and reverse T3 are going to be extruded into plasma. But MIT and DIT cannot be taken into plasma because they're non-functioning hormones. So what is going to happen? They're going to be recycled in the body. And the only way they're going to be recycled is by them being broken down. So we have this enzyme called the iodinase enzyme, usually breaks down MIT and DIT. Now, when you break down MIT and DIT, you're going to form what? You're going to form iodide ions and tyrosine molecules. So that tyrosine molecules that has been removed from this recycling process can be used to form thyroglobulin, while the iodide ions can be taken to form iodine ions, which again can be recycled for the process to continue. So this is basically how we form and we release T3, T4, and reverse T3. Now, for the transport, we have three important proteins that I want us to understand. We have thyroxine binding protein, which is the major one. We have the thyroxine binding prealbumin, also called transthyretin. And we have the albumin. Albumin is the most abundant protein in the body, usually produced from the liver. So the thyroxine binding prealbumin is quite important for this section. Why? Because transthyretin has been implicated in something called amyloidosis. Amyloidosis is when we have a misfolded protein being deposited in the ECF. So we have some cardiac conditions, both senile amyloid cardiomyopathies, and also have the familial cardiomyopathies, which can be due to transthyretin. So it's a very important protein as far as amyloidosis is concerned. Now, this T3 and T4 usually have a very high plasma binding protein capacity. That's very important. And because of that, they are going to have a very long half-life. Usually, thyroxine has a half-life around 6 days because it binds strongly to its transporting proteins, the plasma proteins, while triiodothyronine is usually also having quite a long half-life, but it has a very low affinity for this plasma protein compared to thyroxine hormone. That's why it has a half-life of around 1 day. Now, so this is basically the control of the thyroid hormone secretion. Now we have the hypothalamus. Remember the stimulus for production of thyroid hormones can be decreased temperature. So whenever we have decreased temperature, this will be sensed by the receptors in the hypothalamus, and the hypothalamus will be triggered to produce thyrotropin releasing hormone, TRH. Now TRH will go via the blood to the anterior pituitary gland to stimulate it to produce TSH, thyroid stimulating hormone, otherwise called thyrotropin. And this TRH usually stimulates the thyroid follicles, which are cells that produce TSH in the anterior pituitary gland. Now, this TSH can now go to the thyroid gland, where they usually prompt it to produce thyroxine hormone and even T3 and reverse T3. Now, for us to produce thyroid hormones, we require iodine, and that's why you have iodine here. And remember, this TSH can hypertrophy the thyroid gland. It can make the thyroid gland to increase in size. And that's very, very important when you start talking about the pathophysiology of hyperthyroidism and hypothyroidism. Now, this thyroxine can have a myriad of effects in the body, like increasing metabolism, etc. Yep. Now, here we have something called the negative feedback loop. We have the short negative feedback loop and the long negative feedback loop. So once the T3 and T4 have been produced in copious amounts, you can have a negative feedback loop to the anterior pituitary gland, slowing it down from producing a lot of TSH. This is called the short negative feedback loop. But the long negative feedback loop will go all the way to the hypothalamus, and it will prompt it to reduce the secretion of TRH, especially when you have abundant T3 and T4. So mechanism of action is genomic and non-genomic. What do I mean? It can either be at the nucleus level, or it can even be at the cell membrane level, just affecting some enzymes and some ion channels. So for the genomic cellular effects, you need to have transcription and translation. That's why you talk about a genome, a gene transcription. Now for the non-genomic, usually it's about regulating the ion channels and things like oxidative phosphorylation. So you see that the non-genomic action is usually much more faster than the genomic, because for the genomic, we need to produce new proteins, which will take quite some time. Now, this basically thyroid hormones, which are nuclear receptors. So in an exams, you need to know that thyroid hormones are nuclear receptors because they're lipophilic. So in this diagram here, we have T3, which is making 10% of the thyroid hormones, and T4, which is making around 90%. So we say T3 is most potent. So we need to convert T3 to T4 to T3. And now T3 can come and bind to the nuclear receptor. But if you have T3 alone, it can just come and bind itself to the nuclear receptor. So after binding to the thyroid hormone receptor, we usually have a complex being formed with another receptor called retinoid X receptor, after which now this thyroid hormone can bind to the hormone response element of the gene. Now, after that happens, we're going to induce gene transcription, forming messenger RNA. And from transcription, we usually have translation, where we can now form new proteins. The reason why thyroid hormones must act via nuclear receptor is because they usually entail production of new proteins, especially enzymes. Remember, enzymes are proteins, and thyroid hormones usually increase metabolism in the body. So because of that, they need to really increase gene transcription for them to produce more proteins in form of enzymes. Now, these enzymes can help in different metabolic processes. Now, we've talked about TSH, usually produced from the anterior pituitary gland. And in as much as we have said that the thyroid hormones have nuclear receptors, TSH usually has a cell membrane receptor, which is G protein coupled. And it always acts via something called a cyclic AMP. So this is a G coupled receptor, and this is a cyclic AMP. G coupled receptors are usually serpentine in nature. They cross the cell membrane 7 times. So you see here we have the receptor for TSH, and once it has been bound to TSH, you're going to have activation of the GS coupled receptor, which eventually is going to activate the adenylyl cyclase enzyme. Now, after activating the enzyme, we can now have a myriad of cascade where we can form cyclic AMP from ATP. Now, this cyclic AMP can go and activate protein kinase going downstream. So this is basically the mechanism of action of TSH. Important things to carry home is that this TSH usually acts via plasma membrane receptor, which is G-coupled receptor, serpentine, and it usually serves by increasing the cyclic MP levels and even the protein kinase A levels in the body. So the actions of thyroid hormones are quite easy. It will stimulate carbohydrate metabolism because we are going to increase the carbohydrate breaking of the glycolytic and the TC cycle enzymes, which are proteins. Stimulate fat metabolism. It will have effects on plasma and the liver fats, definitely. And again, it will increase requirements of vitamins because remember some enzymes are usually vitamin B6 dependent. So we need to increase the levels of enzymes. Some of them are even biotin dependent, B9. So we need to increase the amount of vitamins. It will increase the basal metabolic rate because metabolism will generally increase in the body. It will decrease body weight because it has been found that this hormone can even break your muscles. So you are going to lose your body weight. So that's the end of our presentation today. The next video will be talking about the thyroid gland pathophysiology. So you can just go and look at more actions of thyroid hormones, but take home messages that they generally increase the metabolic apparatus of the body.